An action potential is a rapid, transient, all-or-nothing electrical impulse that travels along the membrane of excitable cells, including neurons, muscle cells, and endocrine cells. It serves as the fundamental unit of cellular communication in the nervous system, enabling long-distance signaling without degradation of signal strength. The duration of an action potential in neurons is approximately 1 to 2 milliseconds. All cells maintain a voltage difference across their plasma membrane. The resting membrane potential of a typical neuron is approximately negative 70 millivolts, meaning the inside of the cell is negative relative to the outside. This resting potential is generated by two main mechanisms. First, the sodium-potassium adenosine triphosphatase pump actively transports 3 sodium ions out of the cell and 2 potassium ions into the cell, creating concentration gradients. Second, the membrane at rest is much more permeable to potassium than to sodium due to potassium leak channels, allowing potassium to flow down its concentration gradient out of the cell, which makes the inside negative. The equilibrium potential for sodium is approximately positive 55 to positive 70 millivolts, while the equilibrium potential for potassium is approximately negative 90 to negative 100 millivolts. Several ion channels are essential for action potential generation. Voltage-gated sodium channels are transmembrane proteins with two key gates, an activation gate, also called the M gate, and an inactivation gate, also called the H gate. At rest, the activation gate is closed and the inactivation gate is open. Depolarization causes the activation gate to open rapidly, allowing sodium influx. The inactivation gate then closes slowly, stopping sodium influx, and the channel cannot reopen until the membrane repolarizes and the inactivation gate reopens. Voltage-gated potassium channels have a single gate mechanism and open more slowly in response to depolarization, allowing potassium efflux during repolarization. Leak channels are always open and help maintain the resting potential. The action potential progresses through 3 distinct phases. Phase 1 is depolarization, or the rising phase. A stimulus depolarizes the membrane to threshold potential, approximately negative 55 millivolts. At threshold, voltage-gated sodium channels open rapidly, and sodium ions rush into the cell down their electrochemical gradient. This creates a positive feedback loop where depolarization opens more sodium channels, causing the membrane potential to rise rapidly toward the sodium equilibrium potential of approximately positive 30 to positive 40 millivolts. Phase 2 is repolarization. Sodium channels become inactivated as their inactivation gate closes, stopping sodium influx. Voltage-gated potassium channels open slowly, allowing potassium ions to leave the cell down their concentration gradient. This net loss of positive charge restores the negative membrane potential. Phase 3 is after-hyperpolarization. Potassium channels remain open briefly even after the membrane reaches resting potential, causing the membrane potential to become more negative than resting, approximately negative 75 to negative 80 millivolts. The potassium channels gradually close, and the sodium-potassium adenosine triphosphatase pump works to restore the original ion gradients, returning the membrane to resting potential. Threshold potential is the critical level of depolarization, approximately negative 55 millivolts, required to initiate an action potential. Sub-threshold stimuli produce only local, decremental potentials that decay with distance from the stimulus site. Suprathreshold stimuli trigger a full action potential regardless of stimulus strength. This is the all-or-none principle. The action potential either fires completely or not at all. Refractory periods are intervals during which the neuron cannot generate another action potential. The absolute refractory period is the time during and immediately after an action potential when no second action potential can be generated, regardless of stimulus strength. This corresponds to the period when sodium channels are inactivated and lasts approximately 1 millisecond. The absolute refractory period ensures unidirectional propagation and sets the maximum firing frequency of the neuron. The relative refractory period follows the absolute refractory period and is a time when a stronger than normal stimulus can generate an action potential. This corresponds to the after-hyperpolarization when the membrane is hyperpolarized and some sodium channels have recovered from inactivation. The relative refractory period lasts several milliseconds. Propagation of action potentials occurs through local currents. The depolarized region creates local currents that depolarize adjacent membrane regions. In unmyelinated axons, continuous conduction occurs where the action potential regenerates at each point along the axon, resulting in slower conduction velocities of 0.5 to 10 meters per second. In myelinated axons, saltatory conduction occurs. The myelin sheath insulates the axon, preventing ion flow, and voltage-gated sodium channels are concentrated at the nodes of Ranvier, which are gaps in the myelin. The action potential jumps from node to node, dramatically increasing conduction velocity up to 100 meters per second and making propagation more energy efficient since depolarization occurs only at the nodes. Several factors affect conduction velocity. Larger axon diameter results in faster conduction due to less internal resistance to current flow. Myelinated axons conduct much faster than unmyelinated axons. Higher temperature increases conduction velocity, while hypothermia slows conduction. Higher density of voltage-gated sodium channels at nodes increases the safety factor for propagation. Clinical correlations highlight the importance of action potentials. Demyelinating diseases disrupt normal conduction. Multiple sclerosis is an autoimmune disease that destroys central nervous system myelin produced by oligodendrocytes, leading to slowed or blocked conduction and various neurological deficits. Guillain-Barré syndrome is an autoimmune attack on peripheral nervous system myelin produced by Schwann cells, causing ascending paralysis. Channelopathies are genetic mutations affecting voltage-gated sodium or potassium channels, leading to disorders such as epilepsy from neuronal hyperexcitability, periodic paralysis, and long QT syndrome affecting cardiac action potentials. Neurotoxins that affect action potentials include tetrodotoxin found in pufferfish, which blocks voltage-gated sodium channels and prevents depolarization, causing paralysis and potentially death. Saxitoxin produced by dinoflagellates similarly blocks sodium channels and causes paralytic shellfish poisoning. Batrachotoxin from poison dart frogs prevents sodium channel inactivation, causing persistent depolarization. Local anesthetics like lidocaine and procaine block voltage-gated sodium channels, preventing action potential generation in pain fibers and thus blocking pain signal transmission. Integration with synaptic transmission occurs when the action potential arrives at the presynaptic terminal and opens voltage-gated calcium channels. Calcium influx triggers neurotransmitter release into the synaptic cleft. Neurotransmitters bind to postsynaptic receptors, generating postsynaptic potentials. If these summed potentials reach threshold, a new action potential is generated in the postsynaptic neuron. In summary, the action potential is a rapid all-or-nothing electrical signal in excitable cells generated by the coordinated opening and closing of voltage-gated sodium and potassium channels. The phases include depolarization from sodium influx, repolarization from potassium efflux, and after-hyperpolarization. Refractory periods ensure unidirectional propagation and limit firing frequency. Myelination enables saltatory conduction, dramatically increasing conduction velocity. Clinical conditions including demyelinating diseases, channelopathies, and neurotoxins disrupt action potential generation or propagation, demonstrating the critical importance of this fundamental physiological process for nervous system function, muscle contraction, and sensory processing. Thank you for watching. Don't forget to like, share, and subscribe. See you in the next one.